Dear students, this lecture is about the blood in hemopoietic and lymphoid module. Blood is a specialized connective tissue, consists of formed elements, cells which includes erythrocytes, leukocytes, platelets, and a fluid component called plasma. The volume of blood in an average human adult is approximately 5 liters. Blood circulates in a closed system of vessels. Transport nutrition, waste products, hormones, proteins, ions, oxygen, carbon dioxide, and formed element. The other function of blood is regulates body temperature and assists in regulation of osmotic acid-base balance. The blood cells have a short lifespan, continuously replaced by a process called hemopoiesis. Hemo mean blood in Latin, poiesis mean formation of blood cells to renew the aged blood cells. This is a tube of blood after centrifugation. The precipitate is erythrocytes, while the supernatant is a plasma. In between there is a layer called buffy layer, which includes the leukocytes and platelets. Blood constitutes, as I mentioned, consists of plasma and blood cells. Plasma consists of their composition of water, organic compound, and inorganic compound. The organic compound represented by proteins, amino acids, and hormones, and inorganic compound, salts, dissolved gases, and nutrients. The main component in plasma are proteins, which includes albumin, globulin, and complement protein. Albumin is a small protein, molecular weight maltase 60,000. Preserved the function of the albumin, preserve osmotic pressure in the vascular system, and helps transport some metabolites. While gamma globulin, we have many types of globulins, alpha, beta, gamma. Gamma globulins are antibody immunoglobulins, while alpha and beta transport metal ions as iron, copper, and lipid in a form of lipoprotein. The other plasma protein in the plasma is the fibrinogen. Fibrinogen is a clotting protein, a soluble protein that converted to fibrin during blood clotting. Finally, the complement protein from C1 to 9C represent complement, are part of innate immune system, and they in non-specific immunity, and they function as non-specific host, defend and initiated the inflammatory process. Serum is a part of plasma, yellowish fluid that remain after blood has clotted. It is similar to plasma but like fibrinogen and the clotting factor. Serum is a plasma leaked from fibrinogen and clotting factor. The formed element, the second element of blood formed elements, which includes erythrocytes, leukocytes, and platelets. Erythrocyte اللي هي red blood corpuscle أو red blood cells are rounded in shape, enucleated, biconcave shape, stain light pink. They approximately 7.5 millimeter in diameter. 2.6 is the thickness at the rim, and only 0.75 thick in the center, mean dilated in the center and more thick in the rim. The average lifespan of RBC is 120 days. Aged RBC are fragile and express membrane surface oligosaccharide that are recognized by macrophage. Macrophage in spleen, macrophage in liver, and macrophage in bone marrow, which destroy those erythrocytes by phagocytosis. Carbohydrate determinants for A, B, and O blood group are located on external surface. على external surface تقع receptors مال ال A, B, O on of plasma limي على external receptorات of blood group. Several cytoskeleton protein maintain the shape of RBCs. Cytoskeleton protein give the cytoskeleton to RBC. Mature RBC possess no organelles but filled with hemoglobin, empty of organelles. Erythrocytes also contain soluble enzymes that are responsible for glycolysis, hexose monophosphate pathway, and production of adenosine triphosphate. All these processes occur in erythrocyte that is contain the soluble enzymes that is participate in these processes. This biconcave shape side view of erythrocyte, dilated in the center 0.75 micrometer, while in the rim there is a 2.6 micrometer, therefore appear in stain with a light center and dark margin. This is the erythrocyte, stand one to each other in a capillary. This capillary consists of two endothelial cells. The aggregation of these erythrocytes called Rolex. The hematocrit is the estimation of volume of packed cell volume. Cell represent erythrocytes. Packed erythrocytes, volume, unit volume of blood. The hematocrit is expressed as a percentage. Expressed as a percentage. Normal value are 40 to 50% in men, 35 to 45% in women, 35% in children up to 10 years, and 45 to 60 in newborns. Hemoglobin is a protein composed of 4 polypeptide chains, each of which covalently linked to a heme group. I mean heme in the center, surrounded by a 4 polypeptide chain, or bound by 4 polypeptide chains. The cytoplasm of erythrocyte lack all organelles, but it is densely filled with hemoglobin, the tetrameric oxygen-carrying protein. Tetrameric according to the 4 polypeptide chain that account for the cell's uniform acidophilia when combined with oxygen called oxyhemoglobin, while when combined the carbon dioxide with the hemoglobin called carbaminohemoglobin. The reversibility of this combination is the basic for the protein's gas transport capacity. The second formed element is leukocytes, which is called white blood cells, possess varying number of eosinophilic granules. All leukocytes have eosinophilic granules. The eosinophilic granules are lysosomes containing various hydrolytic enzymes. We have two types of leukocytes, granulocytes and agranulocytes. Granulocytes include neutrophil, eosinophil, and basophil. Possess specific granules with type-specific contents. These cells, neutrophil, eosinophil, and basophil, generate ATP via glycolytic pathway, Krebs cycle. The main cell in Krebs cycle is basophil, while in an anaerobic pathway, neutrophil is responsible for this process that generates ATP. Destruction of phagocytized microorganism by neutrophil occur in two ways, either by eosinophilic granules or by reactive oxygen. The eosinophilic granules release hydrolytic enzymes because it is a lysosome have hydrolytic enzymes to phagosomes to destroy microorganism, or by another way, by reactive oxygen compounds as superoxide, hydrogen peroxide, and hypochlorous acid, formed with phagosomes, catalyzed by myeloperoxidase that's secreted from microorganisms, which destroy microorganisms. The second type of leukocytes is agranulocyte, lake-specific granules with no specific granules, while granulocytes have specific granules in their cytoplasm. There are three categories of lymphocytes. The agranulocytes include lymphocytes and monocytes. Lymphocytes, we have three categories or three figures: B, T lymphocytes, and null cells. B lymphocytes responsible for humoral immune response, يعني secreted antibody against strange antigen that enter the body, while the T lymphocyte responsible for cellular immune response as phagocytosis. The third one is null cells, constitute approximately 5% of circulating lymphocyte and are of two types: pluripotent hemopoietic stem cells, this is the stem cells, and the first type, and natural killer cells. Null cells resemble lymphocytes like their characteristic surface determinants. Therefore, it is called null, because it is like their characteristic surface determinants. This is the types of leukocytes. Granulocytes include neutrophil, eosinophil, and basophil. A granulated cytoplasm with specific granules, fine pink in neutrophil, coarse bright pink or red in eosinophil, and coarse blue or purple in basophil. While the second type is agranulocytes, lymphocytes, and monocytes. The first type of leukocytes is neutrophils, اللي هي polymorphonuclear leukocytes. Consist of 50 to 70 of circulating leukocytes. Their diameter is 12 to 50 micrometer in diameter and blood smear, with a nuclei having 3 to 5 lobes, linked one to each other, these lobes, by a thin thread of chromatin. Neutrophil are usually first leukocytes arrive at the site of infection, where they actively pursue bacterial cells using chemotaxis to remove the invader or their debris by phagocytosis. The cytoplasmic granules of neutrophil, this is the cytoplasmic granules of provide the cell many activity. The eosinophilic primary granules اللي هي lysosomes are large, dense, vesicle with the major role in both killing and degrading engulfed microorganism. The other granules are specific granules. This is specific granules are smaller and less dense, stain faintly pink, and has diverse function, including secretion of various enzymes such as collagenase, delivery of additional bactericidal protein to phagosomes, and insertion of new cell membrane components. The second cell is eosinophil, are far less numerous than neutrophils, consist of 1 to 40 leukocytes. This cell is from 12 to 15 micrometer in diameter as the size of neutrophil, while here the nucleus is bilobed, not as neutrophil, multilobulated. Each lobe attached to another by a thin thread of chromatin. The main identifying characteristic feature of the abundance of large or coarse basophilic specific granules, typically staining red or pink. Ultrastructurally, يعني in EM picture or photo, the eosinophilic specific granules, this is the specific granules, oval in shape with flatted crystalloid cores containing major basic protein, acts to kill parasitic worm or helminthes. Eosinophil also modulate inflammatory response by releasing chemokine, cytokine, and lipid mediators, with an important role in immune defense or an inflammatory response triggered by allergies. These leukocytes remove antigen-antibody complex from interstitial fluid by phagocytosis. Therefore, the leukocyte or eosinophil, the function of eosinophil here, phagocytosis against antigen-antibody complex to remove from interstitial fluid. While basophil, the final granulocytes and leukocytes, also have a diameter from 12 to 15 as in neutrophil and eosinophil, make up less than 100 of circulating leukocytes. The nucleus is divided into two irregular lobes with a coarse specific granules, blue or purple, overlying the nucleus. The specific granules typically stain purple or blue with a basic dye of blood smear stain, and are fewer and larger and more irregular shape than the granules of both mean eosinophil and neutrophil. The strong basophilia of granules is due to presence of heparin and histamine and various other mediator of inflammation. يعني معناها هنا وين تزيد an allergy reaction Madame B, heparin, and histamine as mast cell. When basophil migrate, this is in blood. All this feature and in blood. When the basophil migrate to connective tissue, appear to supplement the function of mast cell. And the basophil, when go to connective tissue, converted to plasma cell. The mast cell and basophil leave metachromatic granules containing heparin and histamine, have surface receptors for immunoglobulin E, secrete their granules component in response to certain antigens and allergens. The second part of leukocytes is agranulated cells, which is represented by lymphocytes and monocytes. Lymphocytes, the most numerous type of agranulocytes in blood smear, which is consists of a family. This is type of family with a spherical nucleus. The smallest leukocytes and constitute approximately 1/3 of these cells. We have two types of lymphocyte, or three types: B lymphocytes, T lymphocytes, and natural killer. Lymphocytes have diverse role in immune defense against invading microorganisms and certain parasites and abnormal cells. Certain parasite against certain parasites as in leukocyte. In immune defense, all the leukocytes function مالتها هي immune defense. We have the small and large lymphocyte. The small lymphocyte are characterized by spherical nucleus. This is spherical nucleus with a condensed chromatin surrounded by a thin rim of cytoplasm. This nucleus occupies almost all the size of nucleus of the size of lymphocyte, surrounded by a thin rim of cytoplasm, making them easily distinguishable from granulocytes, because this cytoplasm with no granulocytes, يعني with no specific granules, but it has eosinophilic granules, while the larger lymphocytes have larger, slightly indented nucleus and more cytoplasm that is slightly basophilic, with a few eosinophilic granules, mitochondria, and polyribosome, and other organelles. The last agranulocyte and the biggest cell is monocytes. This is the monocytes. Monocytes are a precursor of macrophages, and which is called osteoclast in bone, microglia in brain, Langerhans in skin, Kupffer cell in liver, and the other cell of mononuclear phagocyte system. The precursor of all these macrophages that is found in different organs is the monocyte. Monocyte in circulating blood, while when it migrates to the tissue, converted to macrophage. If it is in bone, called osteoclast. If the macrophage in brain, called microglia. Circulating, this is the circulating in a blood smear. This is a blood smear. This is the RBC. Monocyte have a diameter of 12 to 50 micrometer. Have a nuclei of kidney shape or C-shape nucleus. The chromatin is less condensed than in lymphocyte and typically stains lighter than that of large lymphocytes. All monocyte-derived cells are antigen-presenting cells with important role in immune defense. The cytoplasm of monocyte is basophilic. This is basophilic cytoplasm and contain many small lysosomal azurophilic granules. Have azurophilic granules. Mitochondria and a small area of rough endoplasmic reticulum present along with a Golgi apparatus involve in formation of lysosomes. Finally, the platelets. Platelets, this is clump or aggregated platelets of 2 to 4 micrometer in diameter. Non-nucleated, there is no nucleus. Platelets originated from the separation or from the ends of cytoplasmic processes extending from giant polyploidy bone marrow cell megakaryocyte. يعني the precursor of platelets is megakaryocyte. This megakaryocyte giant cell polyploidy located in the bone marrow. When it differentiated, the cytoplasmic processes of megakaryocyte pinches off to originate platelet. Platelets promote the function of platelets is in blood clotting and help repair minor tears or leaks in the wall of blood vessels, preventing loss of blood from microvasculate اللي هي الcapillary and small blood vessels. Normal platelets count range from 150,000 to 400,000 micrometer in cubic millimeter of blood. Circulating platelets, this is, have a lifespan of about 10 days. In a blood smear, platelets often appear as a clump. Each platelets have lightly stained peripheral zone of hylomeric and central zone هي granulomeric تكون more darker in the center and surrounded by a peripheral zone تكون lightly stained. السنتر هو granulomeric والperipheral هو hylomeric اللي يكون more lighter in stain. And thank you for listening.